So the solutions to the first assignment have been posted. I'll be posting the second assignment soon, tomorrow, probably tomorrow. This next assignment you may have 2 weeks on this one because there's stuff that I want to be on it that I may not get to until early next week. So I'll let you know, or it'll be on the assignment when it's posted when exactly it's due, but it may be not this Sunday, but the next Sunday. Assignment 1, you had a few things to do with 2 different genes, SPRR4 and then BRCA1. So first I asked you for sequence information on SPRR4, and you could use the Biomart package to get these. So here's the cDNA sequence, here's the peptide sequence. There was an asterisk at the end of the peptide sequence that was returned to you when you asked for the sequence. That's just a convention. I'm not sure it's used anymore, but it's a convention for indicating the end of a peptide sequence. You could find the entree ID for this gene, so translating between one code type or name type to another. You could Google that entree ID and you get the same information as if you Googled this HGNC symbol. The gene ontology information for SPRR4, there were 2 biological processes, so BP gene ontology terms for this gene. One of them was peptide cross-linking. The other was keratinization. So I don't expect you to know what those are, but those are 2 biological processes that this gene is known to be involved in. Number 2 was about BRCA1 and why is it relevant to breast cancer. BRCA1 is what's called a tumor suppressor gene, which means that when it's present or when it's in its healthy state, it is playing a role that suppresses cancer. With BRCA1, it and BRCA2, which are related genes and their proteins, they're normally present or expressed in healthy breast cells. And what they do is, whenever there's damaged DNA, like you smoke a bunch of cigarettes, you get mutation in your DNA. BRCA1 and BRCA2, their proteins go in and repair the mutation or damaged DNA. Could be not just a mutation, could be a chunk of DNA that's cut out whenever cell replication happens. Damaged DNA, these 2 proteins help it. If DNA cannot be repaired, then these 2 proteins or genes signal for the cell to be killed. So that protects you against cancer in particular, because cancer is a result of mutation. And if there's too many mutations that accumulate and that are not fixed, then that's a process that can lead to cancer. So if BRCA1 itself is mutated, and therefore it doesn't work like it's supposed to, maybe it doesn't express its protein anymore, or expresses a slightly different protein that does not have this function that it's supposed to have, then mutations can accumulate and that can lead to breast cancer. So BRCA1 is known to be a very important tumor suppressor gene in the context of breast cancer. We could find out the Affymetrix probe set ID for BRCA1. I think there's actually 2, and so that's fine if you reported 2. Depending on how you queried for this information, you got either 1 or 2. The way I did it, I only got 1. But definitely this probe set is one of them that is for BRCA1. So if I did an Affymetrix HGU133A microarray, gene expression microarray, one of the numbers that I would get out, an intensity number, a quantification of the activity level of a gene for this particular probe set, so 1 row, 1 column, the spot that corresponds to that row and column, it's that probe set ID and it's querying BRCA1. So I could go find that particular probe set on one of these arrays and I could quantify BRCA1's activity level in an individual's tissue. The KEGG bioconductor package would tell us 1 protein pathway that BRCA1 is known to be involved in. It's involved in more than 1, but the KEGG package only gave us 1. That pathway is called Ubiquitin mediated proteolysis. Ubiquitinization is a process of modifying a protein after it's been translated or created. So a gene is transcribed, transcription, and then into RNA, messenger RNA. And then messenger RNA is translated into a protein. That's the central dogma of molecular biology that we've talked about. Translation then is the process that actually creates the protein. At the end of translation, you have a protein. And one thing that contributes to the biological complexity of organisms is that one protein can be modified or tweaked by various functions, by various mechanisms. One of them is this ubiquitinization. So if you give me a protein, I can, quote, ubiquitinize it, which is a chemical process of modifying with some kind of chemical bond the protein and slightly modify its function. So this particular BRCA1, one thing that it does is it is involved in modifying, post-translational modification of a protein. Here's all the other gene symbols, H, G, and C symbols that are known to be involved in that KEGG pathway. So there's, I don't know how many of them, but a bunch of them. And then the last problem was to go with those genes that are involved in that KEGG pathway to do gene ontology enrichment analysis. We could do that using the top-go package and the run test function. I'll show you my code in a second for how to do that. It's a little bit different than what we did in class. How many Go terms had p-values less than 0.01, 0.001? There were 6,668 Go terms that involved at least one of the genes in our KEGG list. So for each one of those, there would be a test, hypergeometric calculation, a Fisher's exact test, for whether that Go term is overrepresented in our in these genes. Out of all those 6,668 Go terms, we had a p-value for each one of them. 243 of those p-values were less than 0.001. A lot of them were very, very small, like 1e minus 40 or so. Remember that when we are doing this particular Go enrichment analysis, when we use the top-Go package, we don't get any adjustment for doing multiple testing. We just did 6,668 hypothesis tests simultaneously, so the p-values that we get out don't have the interpretation that we're used to when we do a single hypothesis test. So we have to be careful interpreting what these p-values are. Ideally, we would do some kind of adjustment for the multiple testing, or we would translate our tests' significance levels into something alternative like false discovery rate. We'll do that more later in the semester. But for our purposes, a small p-value is indicative of enrichment. So we can order the Go terms based on p-values and look at the ones that had the smallest p-values. Those are probably enriched. The smallest p-value corresponded to this Go term ID. And this was a biological process, not a cellular component, not a molecular function, but a biological process. So high scale or high level function that a gene is involved in. And that biological process was listed as protein modification by small protein conjugation. And if you looked a little further in that description file at the Amigo site, if you Google that Go term, it takes you to an Amigo site that we saw last week. It told you that's the title of it, protein modification by small protein conjugation. So it's protein modification. That sounds like post-translational modification that we talked about with ubiquitinization. And in this help file did say that one of the mechanisms for doing this, the protein modification, is by ubiquitin or ubiquitinization. So that's there to say that this Go enrichment did agree with the KEGG term that we found. KEGG term for BRCA1 said this gene is involved in a pathway that's post-translational modification by ubiquitin. And when I searched all the genes that are involved in that pathway, I found that the most enriched ontology term was a biological process that was basically that. So that makes sense. Here's the code. So I'll always include my R code at the end of the solutions. I'll just show you the enrichment code. So I did a couple of things. First, I got the probe set IDs for all the genes on HGU 133A. And then I went and got the probe set IDs for the 139 genes that were involved in the KEGG pathway. So I need both of those. The total list of probe set IDs was my total collection of genes. And then the probe set IDs for my 139 genes were my selected genes. Those are my interesting genes. So I wanted to go enrichment analysis on those 139 genes that were selected from the original total collection of probe set IDs on that array. In the run test function, we needed a top-go data object. The top-go data object is defined in the line above here. And for defining the top-go data object, we needed a score for all the genes. So for all the probe set IDs on that Affymetrix array, I need some number or character string or something that has an entry for every one of those probe set IDs. And I also need a function that I can apply to that total list of probe set IDs to pull out only the ones that I'm interested in. In this case, only those that are involved in the pathway, KEGG pathway that we're interested in. Right. So the total collection of my genes and a score for them. I could have made this anything. All I needed was something where there was a distinguishing characteristic for the probe set IDs that were in my pathway. So what I did was assign a 0 for every probe set that was not in the pathway and a 1 for every probe set that was in the pathway. So I set up this gene scores object that was a bunch of zeros initially, 0 for the entire length of all the Affymetrix symbols. So for all the thousands of probe sets on this array, put in a 0 in this vector. And then I'm going to put a 1 everywhere that I have a probe set that was in my pathway. So I did gene scores indexed by JJ. And wherever JJ is, plug in a 1. JJ is a set of indices. So I went and got the AffySim KEGG is the list, it's a vector of probe set IDs for all the 139 genes in my pathway. AffySymbol is, is the total collection of probe set IDs for this array. So I'm going to match my symbols of interest in the total list. So JJ is going to be like 5, 12, 13, 42. Those are going to be all the places in the total list of probe set IDs that I can go to find probe set IDs for my genes. If there's 20,000 probe sets on the Affymetrix array total, JJ is going to be the index for all of those that correspond to genes in my pathway. So now when I come here and say gene scores JJ gets 1, that's going to 5, 6, 12, 42, and so on, and plugging in a 1. We needed this vector to have names for each of the elements, and the name for each of the elements needs to be the Affymetrix probe set ID. So that's what I did here. The names of each of the elements is the names of my AffySymbol object. And then from there, it was running the same way we did in class. So go run test on my top Go data object, classic algorithm of the Fisher's exact test. And there we go. So any questions about the first homework? Okay. All right, so we're going to move into new material. This corresponds to the next chapter in the textbook. I'm still waiting to hear back from the library about the issue that I talked to you about. I'm going to give them a full week, which will be tomorrow. If I haven't heard back from them by tomorrow, I'll bother them again. So hopefully we'll get a solution there. Again, you can be 99% covered if you just look through my R scripts, because I'll have similar information throughout our scripts. So the next material we're going to play with is sequence analysis. We talked about sequences in the previous material. How do you download a sequence? What does it look like? What's a peptide sequence versus a genome sequence? We're going to talk more about that in detail. One of the main applications that we'll see here in this section is alignment. So if I have two genetic sequences, how do I line them up in an optimal way? I can make a gap. I can shift one way or the other. But if I have, let's say, a gene sequence for a monkey and a gene sequence that I think is similar to that one for the human being, can I find a way, an optimal way to line those two up so that I learn something about the similarity of the genome between those two species? Or if I have 10 genes from different organisms, say, can I find an optimal way to line all of them up simultaneously? Alignment is a big part of bioinformatics analysis. For example, if I'm sequencing a genome of an organism for which I don't know the genome yet, but I know the genome of a related organism, then if I get little reads, short sequences for the unknown genome, can I learn something about those by mapping them to the genome that I know something about already? So if I have a little chunk of DNA for organism X, whose genome I don't know, but I know that organism X is related to genome Y. They're close to each other in an evolutionary path, for example. Then if I can align that genomic sequence or gene sequence for organism X to the genome of organism Y, and if it lines up in a region that is a gene of known function, then I can say, I can infer, try to infer something about that gene in organism X. That's an alignment problem. When we talk about this important application or technology in omics data these days of next generation sequencing, that's basically really high throughput, really fast sequencing, figuring out the A's, C's, T's, and G's of a chunk of DNA that can be used for a wide variety of applications. It's really important, really widely used nowadays. In the context of next generation sequencing, one application would be, in next generation sequencing, we'll talk about this a little bit today, you get lots and lots of little short reads, sequences, and if I'm analyzing, say, a tissue sample from a human being, and I do next generation sequencing, I might get millions and millions of little bitty sequences from the genome of that individual. I know the genome of the human being. Human Genome Project gave me that. So if I can take these little genetic sequences and map them to, or align them to, the human genome, then I can do a variety of things. So next-gen sequencing, a big application involved in next-generation sequencing is taking lots of little short reads or sequences and aligning them to a known genome or a reference genome. So alignment is a big part of what happens in bioinformatics. It's a huge computational problem. We'll see some of that today, and you'll see it in your next homework assignment. There is statistics involved because not only are we computationally trying to find how best to line up two sequences, but we would also like to put some kind of confidence number on once I've lined up two sequences or multiple sequences with each other, how confident am I in that alignment? So we'll talk about how to put a p-value on an alignment. First, let's start with getting a sequence. We've already seen the Biomart package for doing this. So here's a reminder on how to use Biomart, and then we're going to meet a new package, Seq-NR, which we'll use more frequently moving forward. So Biomart, I library it in. And then I select a mart. Remember that there are different marts depending on the database I want to look at. Ensemble was one biomart that we could use. Once I select a biomart, I have to select a data set within that biomart, and we're going to select the human genome database. So I library it in, and then I select the mart I want to use. Actually, before we go here, we'll finish this real quick, and then I'll do a little more background on sequencing before we dig into the R code. So I use mart, and then I use the get sequence function to actually get the ACs, Ts, and Gs if I want DNA sequence for a gene of interest. So here I'm providing H, G, and C symbol. I have to specify that the type of identifier I'm providing is an H, G, and C symbol, and that gives me this. There's actually a let's see. Why did that look that way? Not used to seeing that output. We'll leave it there. We're going to see the sequence of BRCA1 here using the Seq-NR package. So I only got one sequence. It's here. We're going to get multiple sequences for any one gene. We'll talk about why that is, what that means. There are different variants of the same gene. So talked a little bit about that. One gene can code for a protein, but depending on how you transcribe a gene, you can get different proteins. So there's something called alternative splicing that we'll talk about. In this case, there's supposed to be 30 different sequences for the BRCA1 gene. We'll see that with Seq-NR in a minute. Before we go into the R code in too much detail, we're talking about sequence analysis. So once I have ACs, Ts, and Gs, what do I do with them? Once I have peptide sequences, what do I do with them? How do we get sequence information? It's a bioinformatics task. How do I go through and with a tissue sample, here's a breast tissue sample, say, how do I extract DNA from it and figure out what ACs, Ts, and Gs it is? So I like true crime shows, forensic files, if you've ever seen that. A big application in forensics is if there's biological evidence at the scene of a crime, can we figure out the DNA sequence for who left that there? Sequencing, it would involve that. That's what sequencing is all about. So how does sequencing happen? It used to happen using a process called Sanger sequencing. So this was kind of the workhorse of DNA sequencing up until lately. Sanger sequencing was the prequel to what's called next-generation sequencing that's used nowadays more frequently, next-gen sequencing. But we can learn about the basic sequencing idea in terms of Sanger sequencing. So it was developed in 1977. It's, relatively speaking, very expensive and very slow. It was the basis for the earliest large-scale sequencing projects like the Human Genome Project, which is why that project took so long and cost so many billions of dollars. The basic idea of sequencing, at least in Sanger sequencing, which is the motivation or the template that Nextgen sequencing is built on, is to take a molecule of DNA or a bunch of DNA strands, break them into single strands, and then add those single strands into a cocktail of various things. So a DNA primer. A primer is a short little sequence that will go and stick to your sequence of interest that you can attach to various things. So if I have a single strand of DNA, to do sequencing, what I want to do with Sanger sequencing is add one at a time A, C's, T's, and G's. I want to have the A, C's, T's, and G's fluorescently labeled so that when I add the next piece to this little sequence, the single-stranded sequence, I'm going to add a complementary base pair, or base, to pair up with the next available base on the single strand, and I want it to have a fluorescent dye on it so that when I shine a laser at that single base, I get an image back. Take a picture, and the color of that dye will tell me whether an A, C, T, or G bound at that particular spot. So a picture like this one, if I have a single strand of DNA, like this green one here, that's labeled template, that didn't help much. I'll say I have a single strand of DNA in green here. I have what's called a primer that sticks to one end of the template, and then I'm going to add one at a time A's, C's, T's, and G's to the primer. And each time I add one base, I'm going to take a picture. So for the first base, I'm going to have my primer, and I'm going to add something, one base right there. And when I take an image at that point, it's going to shine a purple light, say. So we're going to have 4 different dyes. Red is for a T, blue is for a C, green is for an A, and purple is for a G. So using chemistry, I add one base, I shine light to turn it on, the light, the dye. If it shines purple, then I say the first base in my unknown sequence is a G. I then add the next base, and depending on what light it shines, I have the next letter. Do it again, do it again, do it again, do it again. And I sequentially build up the A's, C's, T's, and G's for my template strand of DNA. I get an image that looks like this down at the bottom, which is a series of peaks. So going from left to right in this image of peaks, I have location along the strand. So the first peak would be corresponding to the first base that I added, and there's the possibility for multiple colors of intensity, because there will be thousands or millions of copies of my template DNA. So some of the time, just by random chance, a green base will bind even though it shouldn't, or a red base or a blue base. But if it's, what I'm looking for is a consensus at base 1, a peak of a particular color. So if there's a million versions of my template DNA and I do this first base and I take a picture of all the lights, what I should see, if it is indeed a G at that first spot, is a tall peak of purple and maybe some background peaks of the other 3 colors. So I can do a computational analysis to go through all these peaks and take the color, the base, that has the dominant peak and assign that to my sequence. That's how Sanger sequencing works. There is a statistical component to this in that these peaks are random. There's random height. There will be background noise. that can depend on the chemical properties of the DNA strand we're trying to sequence. What we end up with, there is a statistical measure of confidence for every single base that's called. I'll show it to you in a minute. It's called the Phred score, P-H-R-E-D, and it is a lot like a p-value. It's a probability, an estimated probability of an incorrect call at that particular location. So if I'm sequencing a chunk of DNA, 500 base pairs, say, in length, then I get a call at each one of those locations, A, C, T, G, and so on, and associated with each one of those calls, I have an estimated probability. So I don't, in addition to the sequence that I get, I have numbers that quantify my confidence level in those calls. All right, so that's Sanger sequencing. Sanger sequencing is still used. It's particularly useful for longer sequence reads, like greater than 500 nucleotides. Nextgen sequencing is mostly limited to short little reads, which means that for a large genome, we'll break it up into lots and lots of small reads and then try to put those back together. Sanger sequencing still is useful because it can be applied to longer reads. All right. Next generation sequencing is the alternative way these days of obtaining sequence information. So here's information on DNA sequencing generally, and I wanted to show you some information specifically to next generation sequencing. Let's see. Let's look at sequence assembly. So what happens with next generation sequencing is you take a genome and you break it into tiny little pieces, like I said. So millions and millions of tiny little pieces, and then you sequence each of those tiny little pieces using the next-gen technology. So you get to, with next-gen sequencing, like Sanger sequencing, you do one genetic piece, and you sequence that one piece. That's your chemistry experiment for the day. Next-gen sequencing, you can sequence millions of little pieces simultaneously. So kind of like a microarray, instead of just quantifying the activity of one gene like we used to be able to do years ago, now we can quantify the activity of thousands of genes once using the gene expression microarray. Same idea with next-gen sequencing. I get to sequence little things, but I get to do millions and millions of them, or a bunch of them, simultaneously. One of the major applications of next-gen sequencing is the assembly of a genome. So the Human Genome Project was all about trying to construct the entire ACTG sequence of the human genome. There's a bunch of chromosomes that make up the human genome. Can we figure out the ACTG sequence for each one of those chromosomes? If I can, then I know the genome for the human being. The big piece of that puzzle is taking relatively short sequence reads and putting them together into one giant sequence read. We don't ever get to sequence an entire genome because it's too long. We always end up breaking the thing up into smaller pieces. So where was there's some analogies in here that I liked. Right. So the sequence alignment problem or assembly problem can be compared to taking many copies of a book, genome analogous to book, shredding them up, so chopping them up into lots and lots of small pieces, and then looking at each of the little pieces and trying to put the book back together with those little pieces. So we take DNA, the genome, we break it up into millions of little pieces. We sequence each one of those little pieces, so I get the ACTG sequence for each of the little pieces, and then I try to assemble all those pieces back together into a whole genome. That's the computational problem of sequence assembly. Here's the typical scenario. The black sequence corresponds to the sequence in the actual genome that I'm trying to recreate or assemble. What I end up with is a bunch of shorter sequences. So when I broke the DNA up, it broke up at a bunch of different locations. One chunk of one of the copies of DNA had this sequence, according to my Nextgen Sequencing technique. Another chunk of some of the broken up DNA had this sequence. Here's another sequence. Here's another sequence, and this purple sequence actually showed up more than once in my target region. So the assembly problem is taking the purple, a bunch of copies of purple, a bunch of copies of red, blue, and green. I don't know what black is, and trying to line them up so that there's overlaps that make up an entire region. So that's a computer science problem. You can see at a small scale how complicated it could be. You can envision finding overlapping regions like this GCC TGAC in the green chunk overlaps with the GCC TGAG in the red chunk, and similarly these regions overlap with red and blue. A big challenge with assembly is the purple type of read that shows up repetitively. So the shorter the read, the more likely that sequence is to show up multiple times, the same sequence in the genome. So where do you put it in the genome? For this case, the purple read belongs in multiple places in the genome. So I would be incorrect if I said that all of these go here. I'd miss that they go here as well. There are a couple of different flavors of assembly, and you would choose one of these depending on the application. So there's something called de novo assembly, and there's mapping or alignment. De novo assembly is what you would do if you had a Human Genome Project problem, for example. I don't know what the human genome is. I want to take these millions of little sequence reads, and I want to put them together into back together into this book that's been shredded into many, many pieces. That's called de novo sequencing in the sense that I don't have a reference genome that I'm assembling with respect to. I'm just saying there is some genome. Here's all the pieces. Let's try to put them together into something I don't know anything about yet. That's de novo, as opposed to mapping or alignment, where you may have a sequence of an organism that you think is similar to the one you're trying to sequence, in which case you are aligning to that, or with something like RNA-seq that we'll talk about later this semester, Nextgen sequencing application where it's transcription quantification, you have a lot of little reads, and you have some number of copies of each of those reads. 5,000 copies of this little bitty read right here. Where in the genome does that line up? That's an alignment problem. If I'm doing a study of human beings, I get a tissue sample from 20 different human beings, and for each one of them, I do this Nextgen sequencing RNA-seq application. I get counts of millions of little reads. Mapping or mapping or alignment would be here. I know the genome. I know the human genome. I want to just figure out where does my read, where do my millions of reads line up in that known genome. So de novo assembly is putting together an unknown genome. Mapping or alignment is taking these little pieces and lining them up to a known genome. And you do one or the other depending on what you're trying to do with your particular application. A lot of this is computation or computer science in origin, or that's the way we approach these types of problems. How do you optimally put together a bunch of sequences? How do you optimally line up sequences to a known genome? There's a blurb here, and I guess they don't have a citation, but they use a greedy algorithm. So a computational algorithm where you loop through all these fragments, you compute pairwise alignments of all the fragments, and you find overlaps between them. You try to optimize the overlaps. You end up constructing little by little, iteratively, all these overlaps into long overlaps until you have one giant sequence. Like I said, there's a bunch of applications. Nextgen sequencing. There's a bunch of technologies that do next-gen sequencing slightly differently. We're not going to talk in detail about that, but if you're responsible for dealing with some of these data sets, you probably want to figure out which technique is being used, because that will tell you what kind of data you have. There's different data files, there's different data types, depending on what technique or technology is being used. Can't find the section I was looking for, but there are a bunch of different applications of next-gen sequencing or sequencing in general. One is actual genome sequencing. So if I want to know the genome sequence for an organism, I can use next-gen sequencing, or I could use Sanger sequencing, just trying to put together the AC, T, and G sequence for an organism. In next-gen sequencing world, that would be referred to as DNA-seq. So there's several different technologies that are such and such dash seq, S-E-Q, which defines the application type of that next-gen sequencing flavor. You can use next-gen sequencing to find locations in the genome that proteins bind to. So I think I mentioned in a previous lecture this idea of a transcription factor. Transcription factor is a protein, or it's a location in the genome, in the DNA, that is complementary to a protein, or that a particular protein goes and binds to. And one application of that is a gene could be turned on or turned off, turned up or turned down, depending on what its transcription factor is doing. So if there's a transcription factor that's specific to, say, the BRCA1 gene, then when that protein binds to the DNA in that location, it can signal turn on BRCA1 or turn it off or turn it up or turn it down. So it's useful to know for a particular transcription factor where in the genome does it bind. If I know where it binds, then I know something about, I probably can find out where the genes are that it controls. So we'll know there are transcription factors that are known. They're known to play this role, but we may not know all the genes that they control. So there's an application of next-gen sequencing called ChIP-seq, C-H-I-P, chromatin immunoprecipitation-seq, whose purpose is that. So let's do DNA sequencing. Every NGS application is sequencing. It's finding ACs, Ts, and Gs. But there's different ways you can apply it. One way you can apply it is to find out where in the genome a transcription factor binds. So ChIP-seq, you may have heard that, or you'll hear it in your collaborations maybe. Probably the most widely used application of NGS is transcriptomics or transcription analysis. Gene expression microarrays are transcription analysis. That's where we have thousands of genes represented by spots on an array, and I get a number that quantifies the activity level, or specifically the RNA count, how many RNA representatives of each gene are present. If there's a bright light, that indicates that there's lots of RNA for that gene, which tells me that that gene is very active. That's microarray stuff. You can do the same thing using what's called RNA-seq, which again is sequencing, but we take the reads, how many reads there are, tells me about how many copies of the RNA there are, align those back to a genome, and now I get numbers that quantify how active that gene is. So it's different from a microarray, but it's doing the same basic idea. Let's quantify the expression level of thousands of genes at once. And then epigenetics is, there's a bunch of flavors of NGS that are for what's called epigenetics. Epigenetics is the study of how genomic function varies in ways that don't depend on the underlying DNA. So DNA can code for genes, code for proteins that do things, but you can still vary expression levels without actually changing the DNA. So transcription factors can be involved in what's called epigenetics, because if that protein comes and binds to the DNA, it can turn off or turn on, turn up or turn down the expression level of a gene. There's things like post-translational modification that we talked about, where the gene coded for a protein, it did its job, but then there are other chemical reactions that happen after that that modify the function of the protein. So epigenetics is that kind of study, and there's a handful of NGS technologies that are specific to epigenetics. There's metagenomics, which is trying to figure out, like if you have in a lake, you have a smear of microorganisms that are living. So there's a green goo on the top of the lake that you're studying. And in that green goo, there are a bunch of different microorganisms living in a community. And you'd like to know which microorganisms there are and at what relative prevalence are they. Next-gen sequencing can be applied there to try to distinguish which genomes are there and in what prevalence they are. So next-gen sequencing, we'll have a whole section on next-gen sequencing using R and bioconductor for that application, quantitative analysis, statistical analysis. That's my spiel on what it does and what it is. Okay, back to R. Let's get a sequence. We're going to again get the sequence for BRCA1. So we know how to use Biomart. Now we're going to meet a new package. It's called seqnr. I'm going to install it first. So BioClite seqnr. Hopefully we won't have a problem. Looks like there's just one package that it depends on, so it's installing that as well. In seqnr, just like with Biomart, we have to specify a quote mart. With seqnr, I have to specify a data bank. That's its representation of all the different data repositories. If I say choose bank open close parentheses, that's a built-in function in the seqnr package that will tell me about all the different databases I can get. We're going to use a data bank called GenBank. I don't think we've met this one before. It's just like Ensemble, Entree, and all these other ones that contain genomic information. So GenBank sequence database contains a bunch of sequence information. So we're going to use that. These are all the data banks that are available. GenBank is the first one, but there's Ensemble, and a bunch of other ones that we're never going to use, but SwissProt for proteomics and so on. Let's use GenBank, and then let's go download the sequence for BRCA1. Here's how I do it. I use the main workhorse function of seqnr called query. Here I'm going to choose my bank. The main workhorse function for Biomart was the getVM function. The main workhorse for seqnr is query. Remember, I can do help. For all these new packages that we see, I can do help package name. And here's all the functions that are in this package. There's a bunch of them. The main workhorse is query, which is here. So here's the help file for query. Easiest to figure it out by looking at examples. Here's our example for BRCA1. I'm going to do query. List name is the name of the object I want to create, BRCA1. The main thing I need to change is query, the argument query. Query equals, and then I'll show you some more details on this, but what is specified here is the species and the gene symbol. So SP indicates that what comes after the equal sign is a species name that needs to match species names that are available. Species equals Homo sapiens, and gene symbol K equals BRCA1. Gives me the BRCA1 sequence object. There's a get sequence function in seqnr. It's different from the get sequence function in Biomart. So I'm going to run this real quick and then we'll quit. And what I'll show you and talk more about next time is that for BRCA1, I just asked for the sequence. There's a long chunk of DNA that is assigned to BRCA1, but whenever I turn BRCA1 into a protein, any gene into a protein, we talked, I think, about exons and introns. Introns being little chunks in that gene that actually are going to be transcribed. Or maybe we haven't talked about that yet. But there's little pieces of a gene that actually get transcribed. There's other little pieces that get cut out and thrown away. Depending on which of those little pieces for transcription I actually pick out and put together, I can get different proteins. So BRCA1, even though it's one gene, can give rise to multiple variants of the same protein. So whenever I asked GenBank for the sequence of BRCA1, I got 136 sequences. These are all what's called alternative transcripts of the BRCA1 gene. So if I Googled that, it's from the BRCA1 gene, but it's its own little function, or own little protein, or its own little RNA with its own known function. So that's the way we get the genetic complexity of the human genome. There's only 20,000 protein coding genes, but you can get different flavors of all those genes in a variety of these mechanisms, which means we can all be so different like we are. Okay, any questions? We'll pick up on Wednesday. Look for the new homework probably tomorrow.